Do you know that the cell is able to regulate its metabolic pathways efficiently to avoid the wastage or unproductive use of its materials? Today we shall be saying the reciprocal regulation of glycolysis and gluconeogenesis. As a way, the cell effectively manages what is happening time to bring about vital life processes. The cell is able to use hormones like glucagon and insulin, as well as allosteric effectors like fructose 2,6-bisphosphate, citrate, alanine, and acetyl-CoA, as well as energy cytosol molecules like ATP, AMP, and ADP to reciprocally regulate these opposing pathways. Now, looking at the opposing pathways on the board, we see that the major point of regulation is at phosphofructokinase 1, pyruvate kinase for glycolysis, and fructose 1,6-bisphosphate and phosphoenolpyruvate carboxykinase and pyruvate carboxylase. These are major enzymes of these opposing pathways that are regulated to bring about efficient regulation. Now, how does glucagon and insulin regulate these pathways efficiently? Is by influencing the concentration of this interesting allosteric effector, fructose 2,6-bisphosphate. This substrate is produced abundantly in the presence of abundance of glucose in the cell. Now, with the absence of this effector, phosphofructose kinase 1 is almost ineffective. And so whenever the cell produces this in abundance, this enzyme is stimulated and then glycolysis proceeds uninterrupted. This allosteric effector, which activates this enzyme, is also able to inhibit this gluconeogenic enzyme, fructose 1,6-bisphosphatase enzyme, by binding to its allosteric site and inhibiting it. So insulin is able to promote glycolysis at the same time inhibit gluconeogenesis. Now, how does glucagon avert this influence of insulin on these pathways? Glucagon comes in also again by acting on the same enzyme that produces this, is able to act on it in an opposing manner so that the enzyme again, by the reason of the second subunit, is able to convert this allosteric effector back to its original substrate, thereby reducing the influence of this molecule on this enzyme, relieving the inhibition caused by insulin, and thereby promoting gluconeogenesis and inhibiting glycolysis. Now, what about the allosteric effectors that act on this? We've explained this. The next will be citrate. Citrate is the first product of the TCA cycle, which is after glycolysis. So when citrate accumulates in the cell, is a signal that the cell no longer need production of ATP from any of this. And so citrate comes in here and inhibits glycolysis at this point, binding to this enzyme as an allosteric inhibitor and inhibiting this activity. While it also comes to this opposing enzyme, the gluconeogenic enzyme, fructose 1,6-bisphosphatase, binds to its allosteric site and activates it. So citrate inhibits glycolysis while at the same time promoting gluconeogenesis. What about acetyl-CoA? Acetyl-CoA abundance in the cell means that the cell has other alternative fuel supply and so does not need to further break down glucose to produce energy via glycolysis. So acetyl-CoA can inhibit glycolysis by binding to pyruvate kinase, the last enzyme on that pathway, thereby inhibiting it. At the same time, this acetyl-CoA can bind to pyruvate carboxylase, the first enzyme of the gluconeogenic pathway, thereby activating it. So we see it's inhibiting this as I'm activating the opposing pathway. And then again, we have another allosteric effector, alanine. Alanine is gotten from pyruvate, the product of glycolysis. And so when alanine accumulates in the cell, it simply means that pyruvate is no longer being channeled to other pathway, which signifies abundance of energy resources. And so when pyruvate accumulates due to backup, is converted to alanine. And so alanine is able to bind to pyruvate kinase, inhibiting glycolysis. So we see that these effectors are able to regulate this. Alanine only regulates pyruvate kinase. Since the product of this is accumulating, it can come back as a feedback inhibitor to inhibit this pathway. And what about the energy status molecules like ATP, AMP, and ADP? How do they help to regulate these opposing pathways? Now, ATP signifies an abundance of energy resources in the cell, and so because there will be no need for further breaking of glucose to produce energy via the glycolytic pathway, ATP comes in at this enzyme, binds to phosphofructokinase 1 as an inhibitor and inhibits the glycolytic pathway. At the same time, it goes to fructose 1,6-bisphosphatase enzyme, binds to it as an allosteric activator, thereby activating this enzyme, thereby inhibiting glycolysis while favoring gluconeogenesis. What about AMP and ADP? These are products of the hydrolysis of ATP. So they signify an abundance. When an abundance in the cell, it signifies energy insufficiency, the need for the production of energy. That means the need for glycolysis. So AMP can allosterically activate phosphofructose kinase 1, promoting glycolysis while at the same time inhibiting fructose 1,6-bisphosphatase, thereby inhibiting gluconeogenesis, which needs energy that we don't have. Also, ADP can inhibit gluconeogenesis by binding to PEP carboxy kinase, inactivating it, and also binding to pyruvate kinase, inactivating it. So in all, we see that these effectors, we use allosteric effectors or hormones that we see that reciprocally regulate this pathway, do so by acting on both pathways at the same time, but in opposing manner. It's either they are activating glycolysis and inhibiting gluconeogenesis, or they are activating gluconeogenesis and inhibiting glycolysis. I trust that we had a nice time together this day looking at metabolic regulation with glycolysis and gluconeogenesis as an example of how cells efficiently manage resources available to them per time in carrying out the vital processes that sustain life. Until I see you again, it is bye-bye for now.